Hello, and welcome to part 1 of our video guide to MCV 102. Before I begin, I'd like to go over some of the prerequisite classes and knowledge you should have for this class. So the official prerequisite class list for MCV 102 is Bio 1A, Bio 1AL, and Chemistry 3B. However, besides the content in those classes, I would highly recommend you refresh your memory and be familiar with the concepts from Chemistry 1A and 1AL, and specifically in the areas of intermolecular forces, acid-base, and kinetics. Because MCB 102 applies these concepts and uses it to cover a huge amount of content, I'm moving through the content from prerequisite classes rather quickly so we can focus on the content of MCB 102. Now, with that said, we're going to do a brief review of the material that I feel is important for this class. First of all, you should be familiar with the fact that water is an electrically and chemically neutral species. However, water can autoionize and form a hydronium ion and a hydroxyl ion, which is acidic and basic respectively. So we measure the acidity or basicity of a compound using the pH scale, which is a negative logarithmic scale, which means that a change in hydronium concentration by a factor of 10 results in a change in pH of 1. And a decrease in pH represents an increase in hydronium ion concentration, while an increase in pH represents a decrease in hydronium ion concentration. So strong acids would be proton donors that dissociate fully in water, while strong bases are proton acceptors that fully dissociate in water. And here we have a titration graph comparing weak acids with strong acids and weak bases with strong bases. And you can see that the strong acids fully dissociate, and so they initially begin at a very, very low pH compared to the weak acids, which do not fully dissociate. So their starting pH is higher. And conversely, for the bases, the stronger bases have a higher pH because they are better proton acceptors. Now, something that's interesting in biochemistry especially is a buffer. And a buffer system is a system for which there is a certain range of pH wherein the pH is resistant to change. So how do we make these kind of buffers? Well, you're going to need a weak acid and its conjugate base, or a weak base and its conjugate acid. You cannot use strong acids and bases to make buffers. The reason is, in a weak acid or base, because they do not fully dissociate, there will be appreciable quantities of both the acidic and basic forms of the species. So if you add more of an acid or more of a base, in either case, they will be neutralized, which keeps the pH relatively constant in the buffering region. And on a titration curve, the buffering region shows up as the part of the curve that has the flattest slope. So at the half equivalence point of this titration, you indicated by the blue dotted line at 0.5 OH math equivalence, you can see that the slope of the titration curve is reached its flattest point. And at this point, the pH of the system equals the pK of the acid or base used to make the buffer. So buffers are typically most effective in the region that is within 1 pH unit above or below the pKa. So in this case, since the pKa is 4.76, you would expect the most effective buffer region to be between 3.76 and 5.76. Now, whenever you're asked to calculate the pH of a buffer system, always try using the Henderson-Hasselbalch equation, which is the equation on the right. So you can see that the pH is equal to the pKa of the buffer system plus the log base 10 of the acid on the denominator and the base form on the numerator. Now, one way to remember whether the acid or base is on the numerator or denominator is to think about how a base or an acid affects the pH. If you added a base, you would expect that the pH should increase. And so if you want the logarithmic expression to increase, then you would want the base to be in the numerator so that adding more base increases the pH overall. Okay, so now let's move on to amino acids. Now, I hope by now everyone knows that all proteins are just polypeptide chains which are made up of amino acids. And the properties of these amino acids control the overall structure and function of the protein. Now, there are 20 amino acids, and for MCB 102, you need to memorize each amino acid's name, their 3-letter abbreviation, their 1-letter abbreviation, and their overall structure at any given pH, as well as their properties such as hydrophobicity, their charge, and any other unique properties. Now, when I say you will need to know how to predict by pH, what that means is you should be able to predict the protonation or deprotonation of the functional groups on an amino acid at different pHs. So the general rule is that if the pH of the surrounding environment is less than the pK of a functional group, the functional group is deprotonated. The pH is greater than the pKa, the functional group is deprotonated. Sorry. If the pH is less than pKa, molecule is protonated. If pH is greater than pKa, molecule is deprotonated. And if the pH is about equal to the pKa, well then the functional group will exist with some of the molecules are partially protonated and some of the molecules are deprotonated. So you'll have like a mixture of the two. Now, all this memorization for these 20 amino acids does sound very daunting. And unfortunately, there aren't many tips or tricks to memorize them, but I would say you should look at the list at least once every day. Make some flashcards of the structures to help you quiz yourself on the structure of each amino acid. And within a week or so, with some practice, you should be able to get them down comfortably. Additionally, you will also need to memorize pKa values for the acidic carboxyl group and the basic amino group present on every amino acid, and as well as some pKa values for unique side chains present on these specific amino acids, which are aspartic acid, glutamic acid, histidine, cysteine, tyrosine, lysine, and arginine. So I'm going back to this chart. If you can rewind the video earlier. The most important groups of amino acids you need to know are that alanine, glycine, isoleucine, leucine, proline, valine, phenylalanine, tryptophan, and methionine are the hydrophobic ones. And so they will typically be packed into the interior of proteins away from the aqueous environment surrounding them. And then there are the acidic and basic amino acids. The acidic amino acids all have acid in the name, thankfully, so they're just aspartic acid and glutamic acid. And the basic amino acids are arginine, lysine, and histidine. Now, one other amino acid I want to point out is cysteine. And cysteine has a thiol group. And so two cysteine amino acids can be oxidized together to form a disulfide bridge, which is a covalent bond, which helps stabilize a protein structure. So another thing to note for amino acids is that because they contain acidic and basic carboxyls and amines respectively, their structure will change based on the pH of the surrounding environment. So, for example, looking at the carboxyl group in a neutral environment, because the pKa of the carboxylic acid is less than 7, the carboxylic acid will be deprotonated in a neutral environment. And conversely, because the pKa of the amino group is greater than 7 in pure water, the amino group of an amino acid will be protonated. And so there are going to be certain pH values that lie between the pKa's of the carboxyl group and the amino group that will cause the amino acid to have both a positive charge on the amine as well as a negative charge on the carboxyl. And so when this occurs, we call that kind of a molecule a Zwitter-ion, which is a molecule that contains a positive charge as well as a negative charge. Now, another interesting fact about amino acids that you should be aware of is that they do have a pi, which is the isoelectronic point, which is also a pH value at which the positive charges and the negative charges on the molecule add up to 0, which means that it has no net charge overall. So how do we calculate PI? Fortunately, it's relatively straightforward. In this example with alanine, which is a hydrophobic amino acid, its functional group is just a methyl group, which is not very acidic or basic, so it will not have a pKa value. So since there are only two pKa values for the amine and the carboxyl group, we just take the arithmetic average of the two to get the PI. So for this example, you would do 2 plus 10 divided by 2 overall is, so the PI gives you 6. Now, if the amino acid has a side chain with a pKa value, then it gets a little bit more complicated. For example, here we have an instance with aspartic acid. Now, aspartic acid has an acidic side chain, which contains another carboxyl group, and that carboxyl group has a pKa of 3.9. And so, like all amino acids, it also has the carboxyl group with a pKa of 2.1 and amino group with a pKa of 9.7. Now, don't worry about the exact pKa values for carboxyl groups and amino groups. You'll see it vary across different sources, but generally, you should just go with the table that I provided you. So in this case, we need to find the PI. So we want to find the pH for which this molecule has an overall 0 net charge. So what we're going to do is we're going to take the pKa of the side chain, which is 3.9, and average it with the pKa that is closest to it in value. So in this case, we have a pKa1 of 2.1, which is closer to 3.9 than pKa3, which is 9.7. So because of that, we'll take the average of 2.1 and 3.9, and then that gives us a PI of 3. Now, the reason we choose the closer one is because aspartic acid has an acidic side chain, it will also tend to deprotonate and create a negative charge. And so if we want to balance out the negative and positive charges, then we want to have basically a point where all the acidic negative charges, add up to be equal to all the basic charges. And so we kind of want the halfway point between the two acidic groups, pK1 and pK2, so that in total there's about a 1-to-1 ratio of acidic negative charges to basic positive charges. So now let's move on to protein synthesis. So as we all know by now, each amino acid has an amino group, which is called the N-terminus, and a carboxyl group, which is called the C-terminus. So in general, the starting end of a polypeptide will be called the N-terminus, because since new amino acids will always add to the C-terminus of a pre-existing polypeptide, the C-terminus will be the side that is constantly changing. Once an amino acid has bonded to a polypeptide, that amino acid is now referred to as a residue. So during protein synthesis, you can see that the amino group of the incoming amino acid will act as a nucleophile and attack the carboxyl group of the growing polypeptide. Now, this forms a new peptide bond via a dehydration reaction, which results in the ejection of a water molecule. Now, this polypeptide bond appears at first to be a single bond. However, because of the lone pair that exists on the nitrogen atom, it can actually form a resonance structure and interact with the double bond of the carbonyl group adjacent to it. And that gives the single peptide bond more of a double bond nature. And because of this somewhat double bond like nature of the peptide bond has restrictions about rotation, which means that it's not freely able to rotate at any angle it wants. So because of this, the rotation angles around the peptide bond are restricted to being either 0 degrees or 180 degrees. What this means is that the R groups or side chains on adjacent residues are either facing toward each other or away from each other. This rotation about the peptide bond is called an omega rotation. So in this case, an omega angle of 180 degrees indicates that the adjacent R groups are trans or on opposite sides of the polypeptide. So this reduces steric hindrance, which makes this 180-degree angle for omega rotation the most common angle. Now, the 0-degree angle results in adjacent R groups being cis to each other and on the same side of the polypeptide. Now, this, depending on the size of the R group, can result in significant steric hindrance, which makes this conformation much less common and less stable. Now, besides the omega rotation, there are two other kinds of rotations possible. They are the phi and psi angles. Now, the phi angle is the rotation between the nitrogen atom and the alpha carbon. And the alpha carbon is named the alpha carbon because it is in the alpha position relative to the carbonyl carbon. So the alpha carbon and the nitrogen atom define this phi rotation, which is rotation about that single bond. Now the psi angle is going to be the rotation about the carbon alpha and the carbonyl bearing carbon bond. Now that bond is also, like the phi bond, a purely single bond, so it's able to freely rotate, unlike the omega bond. So the phi and psi angles are able to freely rotate and take any value between negative 180 and positive 180 degrees, unlike the omega angle, which can only be either 0 degrees or 180 degrees. However, due to steric interactions, there are a smaller subset of angles that the phi and psi rotations most commonly take on in order to form the most stable polypeptide structure. And that will be covered in the next video, so we will end it here for now.